Hello friends and welcome to Zon Academy. In this video we'll discuss the biosynthesis or organic formation of dopamine within the body. This process begins with phenylalanine. 9 amino acids, including phenylalanine, are not synthesized by mammals. Because they aren't synthesized by humans, the only source of the amino acids, which are called essential amino acids, are dietary. Some amino acids such as tyrosine, which are typically non-essential, become essential because of impaired conversion from precursors. Tyrosine, if in excess, can replace about 50% of the required dietary consumption of phenylalanine. Phenylalanine hydroxylase, or PAH, is synthesized in the liver. Mutations in the gene that expresses PAH will result in PKU, with over 400 known mutations of the gene for PAH causing PKU. With PKU, phenylalanine cannot be converted to tyrosine and the levels of phenylalanine rise. When phenylalanine is in excess, it will often convert to phenylethylamine, phenylacetic acid, and phenylacetylglutamine. PAH is considered a mixed function oxidase. This is because it puts one oxygen into the hydroxyl of the product, tyrosine, and one oxygen into the water, which is also a byproduct. PAH's coenzyme tetrahydrobiopterin, or BH4, donates 2 hydrogen atoms. Its product is called dihydrobiopterin, or BH2. BH2 is eventually converted back to dihydrobiopterin reductase with NADH and H, which donate 2 hydrogens. Dihydrobiopterin reductase is a much more complex process, but the specifics aren't important in understanding dopamine synthesis. Tyrosine is a non-essential amino acid and is synthesized through the hydroxylation of phenylalanine in the liver when it's not being ingested at high enough rates. If tyrosine is being ingested at good amounts, the amount of phenylalanine needed in a diet can be reduced by 50%. Tyrosine is metabolized into other catecholamines, including dopamine, norepinephrine, and epinephrine, or adrenaline, and melanin, as well as other metabolites. The synthesis of DOPA occurs in the catecholamine secreting neurons and in the adrenal medulla. Tyrosine is imported from extracellular space, and tyrosine hydroxylase serves as a catalyst for this reaction. However, it's important to know that tyrosine hydroxylase is the rate-limiting enzyme in these reactions. That means it is the slowest of the reactions. DOPA synthesis is very similar to tyrosine synthesis in that it requires BH4 as a coenzyme. Some areas of the brain have the catecholamine synthesis reactions stop at dopamine, in which case DOPA is converted to dopamine by the enzyme DOPA decarboxylase and the process of decarboxylation. Now, neurologists highly value dopamine synthesis, fully utilizing all of its substrates, synthetic and metabolic enzymes, and byproducts, since the synthesis of dopamine and all of its byproducts are useful for treating Parkinson's disease and many other diseases that we'll discuss in the next video. DOPA decarboxylase is also used as a Parkinson's disease treatment as it reacts with naturally occurring DOPA and L-DOPA. In terms of dopamine's metabolism, there are many different processes that metabolize dopamine in the synapse. The primary method of this is dopamine reuptake into the presynaptic neuron. Another termination process requires 2 enzymes to metabolize dopamine: COMT, which stands for catechol-O-methyltransferase, as an extracellular enzyme, and MAO, or monoamine oxidase, as an intracellular enzyme. Some medication for disorders related to dopamine deficiency will suppress these enzymes, these metabolizing enzymes, to stop the metabolism and increase dopamine activity. It's important to know dopamine almost always metabolizes into HVA, which stands for homophenolic acid. Some studies, however, are hypothesizing that HVA concentration in the cerebrospinal fluid, or CSF, correlate to the neurological responses that the brain has to dopamine. Now, on to the anatomy. There are three long neural pathways that dopamine follows. The first being the nigrostriatal tract. Beginning in the substantia nigra, which is located in the base of the midbrain, the substantia nigra are the large curved black structures that we can see in the diagram below. The red nuclei are above the substantia nigra and receive cerebral outflow tracts, which are signals from the cerebrum to the central nervous system. The upper part of the midbrain is called the tectum. The tectum contains the aqueduct of Sylvius A, surrounded by the periaqueductal gray matter and the superior surrounded by the periaqueductal gray matter and the superior colliculi. Oculomotor nuclei responsible for the third cranial nerves are also located just below the aqueduct. Next is the mesolimbic tract, which starts from the ventral tegmental area. It sends signals to the amygdala and other parts of the limbic system and primarily uses D4 receptors, which we talked about in the previous video. This tract is involved in producing positive symptoms of psychosis. Antipsychotic medication work by blocking dopamine transmission in this pathway, reducing psychotic symptoms. This decreases the activity of dopamine in the limbic system, which can be used as psychosis medication. Last but not least, we have the mesocortical tract, which is found in the ventral tegmental area. This terminates dopamine mainly in the frontal cortex and also terminates it in the cingulate and prefrontal gyrus, overlapping with the mesolimbic system. This likely causes the negative symptoms of psychosis that illicit drugs can affect, and the mesolimbic and mesocortical tracts serve as the neural pathway for drug addiction. So that's important to know. That is it for this video, and I hope you enjoyed it. I'll see you at the next and final video of this video series.